Welcome to the Biotech Startups podcast by Exceeder. Join us as we speak with first-time founders, experienced scientists, serial entrepreneurs, and biotech investors about the challenges and triumphs of running a biotech startup. Gain actionable insight into navigating the life sciences industry in each episode as we explore the business of science from pre-seed to IPO with your host, John Chi. The purpose of the Biotech Startups podcast is to provide general insight into the ever-changing world of life sciences through the experience of a variety of guests. The use of information on this podcast or materials linked from this podcast are at the user's own risk. The views expressed by guests and any employee of Exceeder on the podcast are their own and do not necessarily reflect the views of Exceeder or content sponsors. Any appearance on the program does not imply an endorsement or recommendation of any product, service, or entity referenced in the podcast by Exceeder or by its guests. In our last episode, we spoke with Maneesh Jain about his childhood and how entrepreneurship provided a focused way to make societal impact. If you missed it, be sure to go back and give part 1 a listen. We continue our conversation in part 2, diving into Maneesh's experience bootstrapping a startup, scaling go-to-market at Ion Torrent, and how Cirina leverages the power of a simple blood draw.
And so after Affymetrix, when did you know it was time to leave, and kind of what got you to want to start another company? I know you started Aurifex BioScience.
Yeah. So I was at Affymetrix for about 3 years. And I think, like I said, incredible experience, and then definitely an incredible perspective. Like not only is academic, but pharma, and it's global. And so really just amazing that way. But I think what comes with scale sometimes is the sacrifice of innovation. And that really started to emerge at Affymetrix towards the later part. And that just left me missing something. So once I had kind of learned all these other areas, which are really important for scalability and growth and kind of, essentially getting your real world MBA, then I was like at heart missing something. And it was really just taking innovation and making products and to change lives. So what brought me away from there and to Aurifex was realizing, well, now we have pretty amazing technologies, like, with microarrays and sequencing starting to emerge, but we really haven't addressed key applications yet. So what Aurifex tried to do was look at things like circulating tumor cells and circulating fetal cells. So cells which may be circulating in blood, but if you could enrich those, then you could get a lot of information about the tumor or about the baby. And those seemed like, I think that's a through line you'll probably see through my journey, is the prenatal and the cancer applications have been really important in this field because people care a lot about it. They're really important points in people's lives, kind of the bookends of life, if you will. And you have everybody's attention, and innovation really matters in terms of being useful. So I think that's partly why those two have been so front and center in the development of genomics. But with Aurifex, we found a way. The problem was when you capture those cells with high sensitivity, you have very poor specificity. You capture a whole bunch of other cells. And so it's not very useful. Whereas if you try to be very specific, then you miss most of the cells, and they're kind of rare. So you can't really miss most of them. So it was kind of this dilemma that had plagued the field. And this technology also out of the Genome Center, which my co-founder had developed, that was able to do both high sensitivity and specificity. So we got that going. Now, it was 2008.
I was going to say, I see a theme there too, where it's like you had 2000 and then 08, this you full cycle.
Yes, indeed. So it was definitely adversity. And I think what we realized is that we needed to bootstrap this one and just do it as much ourselves as possible. It was going to be impossible to raise venture money for this, or at least in a way that was reasonable for founders. And so we did mostly try to bootstrap this company. And it turned out that Illumina was doing a bunch of pharma projects, and this was going to be a fit for what they were doing. And so it was a pretty early exit, maybe too early. We might have, in different times, I think we might have waited longer and venture funded it and grown it to a much bigger company. But I think that's it was a pretty early stage exit. But what came out of that was really, I think, in my mind, this transition from, okay, these technologies are very cool, but now we can actually start to do things we all really care about, finding circulating tumor cells, circulating fetal cells, and saying more about your cancer if you are diagnosed, or about your baby, and that people is really important information. So a pretty good experience from that perspective.
That, I mean, like what a, I mean, for '08 and during the '08 recession, I mean, even having an acquisition and bootstrapping it is a feat in itself. So I think, that's an incredible outcome. And for, you mentioned that Illumina was kind of in your adjacency. How did that merger acquisition take place, and what led up to that?
I think the initial introduction was because one of my founders at Parallele, Mustafa Ranagi, was now at Illumina. He had started another company, Avantome, which became part of Illumina. And so that was how we had the initial introduction. But I think learning more about their services business and their pharma customers who really wanted to isolate and identify these type of cells, so then they could use Illumina technology on those cells to do kind of profiling, genomic profiling, was a pretty good fit. So it was kind of a sample prep platform for Illumina to use their genomic profiling tools on. So that was a pretty good strategic fit.
I was like thinking too, like it kind of like, I'm just like thinking, like putting it like kind of for now, just kind of like seeing the journey, going to the talk, and then joining the Genome Center just kind of had this Cambrian explosion of just like, this incredibly exciting paths that you can go down. And I'm thinking of like what I can take away from it from in my own experience too, is just like, don't be so shy, John. Like go out there and get exposure and go experience what, get out of your comfort zone. And, you were in physics, but then you went into life science, and not be, too hung up on not having the prerequisite background. Because I think, who could make that call that someone that you worked with at Genome Center would end up at Illumina, and then boom, it like comes like full circle again. So I'm like trying to like, I think I can definitely take home something from that too.
It's increasingly important, I think, for the next generation, right? So for us, it was a leap of faith to kind of switch, and it was a pretty big risk, and thankfully it all worked out, and it's been an amazing journey. But I think be willing to take those risks, and I think as we look at younger people now, and certainly the next generation, our kids, I think they will be switching fields quite frequently, and so the ability to have the confidence to learn something new and apply what you know before to this area, and then expand. That is a pretty core skill set, I believe, overall. So I've done some of it, but I think increasingly there'll be more people doing a lot more of it.
Absolutely. So after the Illumina acquisition, I know you made your way to Ion Torrent, and I know Illumina, Ion Torrent, we're battling head to head. How did you make your way over to Ion Torrent?
Yeah, so that really came about because the head of sales at Affymetrix, Greg Fergus, he had left, I think, around the same time as I left Affymetrix, maybe a little bit before. He'd been there from the 90s and built the company from zero to like 400 million or something like that in revenue. And so he was looking for the next exciting thing. And so we both got introduced to Jonathan Rothberg. And he had this idea, which was, of course, his claim to fame was he did the first next-gen sequencer with 454. But he had this idea where in the life sciences, we've always built our own supply chains. So we build it all ourselves. Whereas when you look at people doing work in the semiconductor industry, or you look at apps, people leverage either, open source code, or they leverage fabs like TSMC and other semiconductor fabs. So they leverage these existing supply chains and they sit on top of that. So it allows them to build applications much faster. And so his idea was, well, instead of building our own supply chain, which again, Affymetrix built their own factory from scratch to do literally from scratch, every bit. Illumina built all the oligo stuff and all their sequencing stuff from scratch, right? Built the whole supply chain, and it takes about a decade to do that. Really well.
And a lot of capital,
a lot of capital. And so the idea that was really compelling here is we knew sequencing was a killer application, but could we leverage a supply chain to sit on top of, in this case, a semiconductor supply chain? And so essentially do sequencing on semiconductor wafers. And the advantage would be is much lower cost. And it turned out also that much faster turnaround time. And so the problem we were trying to solve then is that sequencing in that day, 2009, was these big floor standing devices, instruments, which are like $1 million devices.
I remember those. Yeah.
300 to 400 labs around the world who could afford one and could then actually run it, have the expertise to run it. But many, many tens of thousands of labs wanted to do sequencing, and they didn't always want to do the whole human genome. They sometimes just wanted to do targeted sequencing and get some information on their project. And so I think the specs for Ion Torrent, what made it exciting as well, we could build a benchtop device for close to $50,000 instead of $1 million. We could have the turnaround time, which is about a day instead of a week. and we would deliver more targeted solutions, not the whole genome, at least initially. And that felt really exciting and worth the leap. So decided to jump into Ion Torrent. When I first joined, I think we could sequence, we wanted to do the human genome as our eventual goal, but I think we could do half a base initially.
Wow. So
Because
that was quite a leap of faith. That was quite a leap.
We did C's, but G's and T's had some noise and it wasn't quite. But again, I think we knew that if we could solve the problem to do all the 4 bases really well, the semiconductor platform would scale very readily.
Yeah.
So big leap of faith again, but I think it felt like a technology in the right direction. And of course, having Jonathan Rothberg having built 454, that was promising, is like he knew a thing about sequencing technology. And so we kind of knew the application space was pretty wide open for a benchtop device that could be rapid sequencing in a more targeted application. And the technology felt like a good scale. So yeah, a little scary for the first year there because we had a lot of big milestones.
Yeah.
But then things started to really hum along. And then we kind of, I think the early 2000s were the heyday, 2010 to 2012 was really just a period of very rapid growth for the company. And we grew to about 400 people and had a lot of success in the market. So that was very exciting to kind of take this whole cycle of sort of technology, from technology development to early market development to then, I think, broad adoption in the market was super fun.
There's so many directions I want to go there, but I guess, like, just to go back to your, like, this leap of faith. I know you were the first commercial hire there, and what was it like outside of like product development to like scale up a team of you mentioned like 400 people and then design like a go-to-market and all that? Like, was that kind of like your crack? Like, I mean, you obviously at Affymetrix had your kind of like portable MBA, and is this where you're like, okay, it's time to put this MBA to work? Like, I'm going to, what was your experience as a first commercial hire, I guess, is what I'm asking.
Yeah. So that scaling was over 5 years, right? So it wasn't overnight. At first, we were super thoughtful. So we had, I think, I want to say 20-ish people, 18 to 20 in R&D when I joined. And so the first thing was kind of say, okay, let's point the technology in the right direction. What's the product roadmap? You have a technology roadmap, which is great, but what is going to be the product roadmap and what are going to be the key applications? And that led to some interesting choices, right? At first we were like, well, we can do microbial sequencing. Microbial genomes are small. And since initially we have a lower capacity chip, that's the perfect application. And that was great. But then while we were doing market research on that and talking to early customers, a lot of them came back and said, well, I want to do targeted sequencing of human genomes. Like, so microbial is great, but what if I could do 100 exons, right? Or 10 megabases of sequence that I could sequence more deeply, the regions I care about, because I want to look at mutation profiles. So that took us down the road of saying, okay, that actually is going to be the killer app. Because as we started to bring the microbial sequencing to market, we kind of realized all this demand. And so we actually had to do multiple inventions to serve that. And this kind of gets to the whole product solution. Like, when you're solving a problem, it's much better to solve one problem completely than to partially solve all problems.
Yeah.
So having a sequencing platform that is low cost and a rapid turnaround is partially solving all problems.
Yeah.
But saying, hey, for targeted human sequencing, we have a complete solution. So we needed a prep upfront that would go human sample and produce the libraries for sequencing. And we needed data analysis on the back end that would tell you about mutations and accurately quantify those. So the front and back end had to be kind of pieced together. So that was a lot of innovation. And then I think, again, as we looked at the market, the commercial opportunity turned out with Life Technologies because they had a great sales team in this area. And we were like, well, we could build our own or we could partner with them. And that seemed really promising. That was also classic because the CEO then, Greg Lucier, had, I think, great mindset of saying, well, we don't just acquire technologies, we also acquire people. So they actually left Ion Torrent alone for about 2 years. We had a R&D budget, we had a commercial deliverables like revenue and budget, but they were like, you can recruit from within Life Tech the people who you think are going to be most helpful, as long as they want to come and join you guys.
Yeah,
you guys will get this budget to do R&D, and then you have to deliver these things. And beyond that, it was fairly independent, and that was fantastic because what it allowed us is, through this early stage of the company, really keep the team cohesively together with very clear goals, but working as a kind of startup on steroids, if you will, right?
Yeah,
More resources and a bigger sales team, but still that startup culture. And that's pretty powerful. So I think that's one little bit of advice when it's possible, not always possible, but in M&As, if you can try to keep that team together as part of the contract, so that the team can have deliverables that make sense to the bigger company, but can really stay cohesive with expanded resources, that can really help get that cycle of the full potential of the technology. Because often what happens is companies will acquire companies for a product, and then they put people into all these different units. And so the culture is broken up and the technology doesn't progress as fast, and people wonder what happened. And what actually happened is you broke the will to push the technology to its limits and achieve its full potential. And so that's kind of important to not do that. Unfortunately, it happened.
And I guess when you were, you mentioned this is something that, as you're kind of exploring an acquisition or a merger, is that something you need to set out in the contract? Is this a tall ask of a company when you're joining a larger company, or is this something that is fair game, something that is a discussable kind of term of an acquisition to kind of keep this culture intact and kind of remain autonomous?
Yeah, I think it depends on what the founders and investors want, right? If they're just looking for an exit, then you really can't make asks like that, because then you're looking for the buyer. But I think if you're looking to have the maximum impact with their technology, and the partner is a great partner because they have the commercial resources and they just have additionally resources and they really believe in this piece, then it's a very reasonable ask, because it's a no-lose proposition in some ways, right? I think what would be not reasonable for the buyers to say, well, give us a bunch of resources and we'll see how things do. Right?
Yeah.
That's not reasonable.
Yeah.
But to say, here are the resources we need to bring this to its full potential, and here's the commitments we make on revenue or other things, and here's what we need from you guys to do this, then I think it can be a pretty good win-win situation. And so over time, I think it'll be inevitable that bigger companies will want to fully integrate small companies. But I think having that period where it can realize its potential, that's really key. And so I think if your heart's in the right place, which is I want to maximize the potential and make this available to as many customers as possible, then I think you can find that synergy.
Absolutely. And I think I've had, just knowing that you can have that conversation for anyone, sometimes you don't, if you don't ask, you don't get. Yep. So, I think that's like something that is always worth remembering too. It's like, don't be, they can say no, they reserve the right to say no, but it's always worth asking. And obviously when you ask, have a well thought out formulation of why you're making the ask. And it sounds like, that, and it can, magic can happen. And, you can have the kind of the war chest of a Life Technologies to kind of go forward with Ion Torrent. And so after joining Life Technologies, you mentioned that, Ion Torrent was independent for 2 years. After that 2-year mark, was that when you decided to leave Life Technologies and join Butterfly Network?
Yeah, exactly. So it was after that, we started to integrate Ion Torrent much more fully into Thermo. Integrate into Life Tech, but also Life Tech was then going through this acquisition, which became part of Thermo. Yeah. So when it finally became part of Thermo, then, which is a 50,000 person company, then I was like, okay, I think I have contributed the most I can.
Yeah.
Brought it to as far as I can. And this has gotten to be a much bigger environment, right? Which others are probably more capable at that point. So then that was time for me to leave and made that transition. This time was a little bit easier. I think, the first couple of times you do that, it's really hard. It's like, what the heck am I doing? And what am I going into? I know what I'm leaving, but I don't know exactly what I'm going to, which can sometimes be hard. But I think at this point, I saw much more clearly where the value was and what made sense for me. And then I think I was also, now we had gotten much closer to clinical applications. So one of the most exciting things with Ion Torrent was we built a clinical platform for sequencing the cancer hotspot genes. And this was like, you would get the mutation profile for the key genes, and actually some CLIA labs would use that to make therapy decisions for their patients. And we would actually get letters from patients whose course of treatment was altered based on the profile, and it changed their life, right?
Wow.
Or saved their life. And so I think just to get even a few of those letters is incredible. Like you just kind of feel, oh, wow, we are actually impacting. Doesn't matter how many, but even if it's one person and one letter, and you have made that kind of impact to somebody's life, that's really meaningful. And so wanted to continue that sort of theme of clinical applications. And then it was impossible to turn down Jonathan's next idea.
Yeah.
Now that the semiconductor supply chain has worked so well for sequencing, let's see what else we can use it for. And this time was ultrasound. And so instead of doing sequencing on the chip, we were doing ultrasound on the chip. So integrating what are called these MEMS devices on the electronics of a chip. And the idea there was, again, while there's a sort of theme of democratizing technology here, right? So while ultrasound scanners are available in hospitals, they're really a shared resource, like in emergency departments. And often if a scan is going on, you got to wait for somebody else to get to use it. And sometimes you might have an urgent situation, or you might be in the field where you need this, or a rural area where you may not be able to cover such an important shared resource, and you just want an individual device. So the idea that you could make a simple handheld device that would connect to your iPhone for the readout, right? Again, sort of the old days of Parallele using a assay and using the Affymetrix chip for a readout. Here we were like, okay, we'll develop the device, but then we'll just use the iPhone for a readout because they have all this great capability and screen and apps. And why would you not just leverage that whole supply chain? So that was the idea with Butterfly. It was in Connecticut. And so it definitely made it a little harder to hire people, especially when you move from the research to the more development stage. But we, I think, had a pretty compelling mission, and were able to get some good people.
That's awesome. Did you end up moving to Connecticut, or were you living the bi-coastal life?
Yes, bi-coastal.
Oh,
that was not pretty.
Yeah.
Definitely.
At least you got the frequent flyer miles. So
Exactly.
Yeah, you're.
But yeah, that was, I think, one of the takeaways from that. It was really successful, but I think by the time we were getting our FDA approval and things were going pretty well for the company, and we raised a good round of financing and a great roadmap. It was definitely starting to have an impact on my family. And I just remember this one time where we had just gotten back from a trip from India, and my daughter had been having this kind of stomach ache, and most people thought it was some GI stuff. You usually get a bug when you're traveling internationally, third world countries, whatnot. But it turned out it was her appendicitis. Her appendix had actually ruptured. And when I was out on this trip to the East Coast, and then my wife took her to the hospital, and they were like, "This is pretty bad. We just caught it barely in time. She another day or two would have been septic." And so I think that was, at that point, I was like, I think I need to be closer to my family.
Yeah.
Personal life.
Yeah.
This is not all. It all is not adding up. And so I think I had to make the big decision to transition there. But that was a really exciting journey. And I think it's been, I think there's 50,000 devices out there now.
Oh my God.
Everywhere from the US and rural areas to Kenya and so many countries. So it's really exciting to see it being used so broadly.
That's amazing. And I know, you guys got it cleared for, 13 plus clinical applications with FDA. What was your experience, going through that? That sounds like a lot of regulatory, kind of work that had to be done. What was your experience working with the FDA on that?
The good thing with imaging is that it was a 510K. And so the good thing with imaging is there have been so many predicates that what we were doing is we weren't changing necessarily the imaging was not novel. We were just doing it in the form factor that was very convenient and much more cost effective. And so while it's never easy to go through a regulatory process, it was reasonably well-charted course. So it was a matter of time in doing and producing the data, but not a huge amount of risk in terms of getting the approval.
That makes a lot of sense. I can see like a theme of, the kind of the semiconductor fabs, and then the microarrays. There's kind of like, not to say that like starting from scratch is, there's definitely value to that, but also there's also value to kind of having something that's kind of established and then being able to take it to the next level and scaling it to the next level, which, kind of, again, de-risks it. And as, sounds like the travel East Coast to West Coast was becoming, more problematic on you. when did you just, and after that, when did you decide to join Cirina? And I can see a theme too. And for those who are unfamiliar with Cirina, could you also kind of just like let folks know what Cirina's mission and focus was and kind of what you guys sought out to do?
Yeah. So I think both Cirina and now Mirvie, which we'll talk about, have this are leveraging liquid biopsies fundamentally. And I think this is just so transformative for the field, because as we've gotten to such high resolution in sequencing and counting DNA or RNA, what we've realized is that instead of being invasive and having to get that information from a tissue, which is always what you had to do in the past, now you can get that just from a blood sample, because the blood is circulating through all the cells in your body and sampling all your tissues. And so if you are sensitive enough, then you can pick up information on what's happening in different parts of your body simply from a blood sample. And that's very powerful as a paradigm shift, because now the clinical workflow is really simple, just a blood draw, right? And the information you're getting can be life and death. Or can actually change the course of a baby's development or your cancer's development. And so really powerful information that's available from a simple blood draw. And I think that concept is here to stay. And it is a lot of work to try to get to the levels of sensitivity and specificity you need from just a blood sample, because after all, it's sampling your whole body versus a biopsy is picking up something very localized. But that, I think, was the key concept there. So I think just continuing the theme of clinical applications in genomics, Cirina was very exciting, because I think the idea for early cancer detection actually had come up pretty early. So when we were doing Ion Torrent, there were a lot of labs doing a non-invasive prenatal test. So testing for the baby's Down syndrome, essentially, if the baby had Down syndrome, by looking at the chromosomal abnormalities in a blood sample. And what they found in those early studies is that 1 in 1,000 of the pregnancies, they actually found aneuploidies, not just in chromosome 21 for Down syndrome, but they saw a lot of the genome was pretty screwed up. There was massive duplications or copy number changes, other things in the genome. And so at first they're like, this is some kind of error, like what is going on? And as they looked at the data a bit more, they realized we are actually detecting cancer, because 1 in 1,000 roughly of people, younger people even, will have risk for cancer. And so that led to the idea that, oh, wow, if you can take a blood sample and detect the earliest signs of cancer, can we just do early cancer detection? Because I think a lot of work, Bert Vogelstein and others have shown that cancer depends for different cancers, but they usually develop over the course of a decade or more, maybe 15, 20 years. It's not overnight. And so when you end up getting symptoms, of course, it's progressed to stage 3 or 4, and that's typically when you end up having symptom-based diagnosis. But the development has taken many, many years. So in principle, if you can intercept it in the earlier stages, you can do surgical procedures or something much simpler to ideally be curative, and it hasn't spread, so it's very localized, and you can kind of cut it out, so to speak. So that potential was very exciting. And so Cirina really started because one of our investors at Ion Torrent, Min from Decheng Capital, he had been really fantastic to work with. And also Dennis Lowe, I'd known about from all the works with non-invasive prenatal test. And we kind of pulled this thing together to get the company going. I think they had kind of created the idea, and then we actually needed to start the company and have people and actually progress the technology. So I was founding CEO, and we got the company going with the idea that we could build the first test for early cancer detection.
Very cool. And I know Cirina was also, speaking of travel, I know they were also you guys had also operations in Hong Kong. What did you also have to travel and do that as well? And what was it like building a team kind of, on kind of opposite sides of the world?
Yeah, it's kind of ironic, isn't it? After I basically said the bi-coastal lifestyle doesn't
let's make the coastal even larger coast. Yeah,
do something a bit more international even.
Yeah.
No, I think part of it was the opportunity was really compelling. I think the ability to do blood-based cancer detection, early cancer detection, is just really paradigm shifting, right? So it was very compelling. And then actually we built the team first in the Bay Area. So
okay,
the Hong Kong team was because we knew that the first trials we had done were in Asia, and the first cancer we were targeting was more prevalent in Asia. And so the CLIA lab we were going to build in Hong Kong. And so a lot of the R&D work was based in the Bay Area. So the early days of the company was really Bay Area based. And then as we got ready to go to market and build a CLIA lab, then we also built a team in Hong Kong. So Very that cool. was not every, I was probably there once a month. And so it was still travel, but not quite
as frequent
bicoastal.
Yeah.
But that was very exciting. I think, it's you just see a lot of emphasis on cancer in the US, but of course we know that it's a global challenge for everybody, and there are different cancers that are prevalent in different areas. So I think that was one perspective. Building a startup was quite different in the two places. In the Bay Area, you had many people, I think, who had been part of startups and seen how their equity value could grow over time with startups, and they would value that piece. And so, a lot of people would say, hey, we know we need to be capital efficient in the startup. I'll take a pay cut, but I'd like a little more equity as much as possible. And so that was the typical conversation in the Bay Area. In Hong Kong, they had been less experience with successful startups. And so the typical conversation was, well, how much pay raise can I get to make this transition, which automatically meant much less equity. But that's, I think, just a sign of what people are used to and like what successes you've seen. And so I think building the teams was pretty different in the two places.
Yeah, sounds different.
But of course, really kind of in terms of mission, we were completely united and people were highly motivated in both Hong Kong and the US. And we did have people go back and forth to kind of keep the synergies. But it helped that we had a focus on R&D with the Bay Area team and the focus on the CLIA lab with the Hong Kong team. So the skill sets and kind of the responsibilities were deliverables was slightly different for both the teams.
Absolutely. And as you look back on your experience at Cirina, what would you say were like some biggest, challenges and triumphs that you experienced during your time there?
I think it was just we were very ambitious. So I think that's both exciting and challenging. So we had a lot of data on one particular type of cancer, but obviously we wanted multiple cancer types, early detection of multiple cancer types. I think some of the challenges were really just kind of thinking how you do these clinical studies globally.
Yeah.
There had been a lot of work in Hong Kong on the studies, and how do you expand that to the US, and how do you think about two indications, and how much of that you can take on as a small company versus a bigger company. And ultimately, I think that's really what led to the GRAIL acquisition decision, was to realize that to do these trials in the US, as we wanted to expand, they would be large trials, and they just needed a lot of capital. And GRAIL was well capitalized, and we had proven technology to do early cancer detection. So that seemed like a great fit. But yeah, I think it was the excitement and the challenge of doing this. But it really, I think, brings you back to how do you advance the standard of clinical care? You need large, definitive clinical trials that are very rigorous, and that's pretty expensive. And so I think that's where it's interesting increasingly to think about how innovation will impact clinical standards of care and how we can do trials more efficiently over time. Because that, I think, does end up being one of the bottlenecks for a startup to think about doing studies that are properly sized and scaled.
Absolutely. And I think it's kind of like the elephant in the room. This is that, as you embark on that journey, it's like the goal is to get to the clinic. Yep. But then when you're there, you're like, oh my goodness, this is such a large endeavor, like in all facets. It's like, how do we go about doing this? And so I'm super excited to see, work being done on how to make that more efficient, reduce the kind of like, the activation energy needed to get it through the clinic, and seeing everyone kind of find ways to kind of improve on that process. Because I absolutely do think it's a bottleneck that hopefully we can alleviate over time.
That's all for this episode of the Biotech Startups podcast. We hope you enjoyed our conversation with Maneesh Jain. To learn more about his journey, tune in to part 3 of our conversation, where we cover the driving force behind founding Mirvie, Maneesh's thoughts on the maternal health crisis, and his approach to building a holistic company culture. If you enjoyed this episode, please subscribe, leave us a review, and share it with your friends. Thanks for listening, and we look forward to having you join us again on the Biotech Startups podcast for part 3 of Maneesh's story. The Biotech Startups podcast is brought to you by Exceeder. Don't want to miss an episode? Make sure to search for Biotech Startups podcast in Apple Podcasts, Spotify, and Google Podcasts, or wherever you get your podcasts, and click subscribe. To learn more about our leasing program, visit our website www.excedr.com. We provide research labs with equipment leases on founder-friendly terms to support a path to exceptional outcomes. On behalf of the team here at Exceda, thanks for listening.